people from the core bioconductor team present with us today. The aim of the whole Bioconductor series workshop, which has been organized by the Asia Africa Coordinating Center, the Bioconductor Community Organization, and Ncbi, is to enable us to grow a community of bioconductor users within Africa. So in order to do that, the Bioconductor community, international community, is giving a series of specialized workshops specifically for African students. And we do encourage you to please sign up to the mailing list, join the groups, and get involved in the discussions and stuff, as it would be great to have more African representation within the actual international bioconductor community. So to get started, we're lucky to have two extremely talented R programmers and core members of the Bioconductor team. The first one is Laurie Shepherd. She's a senior programmer in the Bioconductor core team and started education in biology and chemistry, but decided she needed more computational skills in order to be competitive in the industry. So she switched degrees and got a bachelor's in bioinformatics with a minors in computer science and computer theory. And she's completed her undergraduate degree and started work in the biostatistics department at the University of Buffalo and Roswell Park Comprehensive Cancer Center, where she has been. And she'll be one of our trainers for today. The second trainer we have, which we're very lucky to have on board, is James McDonald, who's a biostatistician at the University of Washington and the Department of Environmental and Occupational Health Sciences. James has over 15 years of experience in analyzing a wide range of high-throughput genomics data that includes microarray, RNA-seq, genomics, SNP, CHIP, and methylation data. He also has an extensive background in programmatic aspects of data analysis, and he's a core member of the Bioconductor project, which is an open source project based on R. So thank you today to Laurie and James, and if you'd like to get us started for today's workshop. Thank you.
Can you hear me?
Yes, we can.
Great. So if everybody's got your orchestra spun up, so it should look like, let me share what I've got. So once you get your orchestra spun up, it should look like this. It should look like an RStudio instance. And remember, the password and the username are both RStudio lowercase. Once you get the RStudio spun up, you should type library see 2021 Anno, and that will launch all of the packages you'll need for this presentation. And it should probably make this larger, which is
Tools global option.
what is it?
If you go to Tools,
Okay.
Global Options,
Okay.
Appearance.
How's that? Is that better?
Yep.
Okay. So anyway, let me switch to you should also open this page. This is what we will be using to follow along with. This is the launch page, which is not actually what we'll be using. So once you get to this page, you click on Articles, and then the Bioconductor 2021 Annotation Workshop. And that will bring this up. Does anybody have any issues with that so far? Looks good. Okay, so I'm James McDonald, and James McDonald, and today we'll be talking about the Bioconductor Annotation Resources. So So the prerequisites that we would sort of hope you would expect that you would know are basic knowledge of R syntax. We're not going to be teaching anything about R or how you do things in R. And hopefully you have a basic understanding of the annotation sources that you can get from, for example, Ncbi or Ensembl. Some background reading that if you haven't read, you might take a look at is the Annotation DBI vignette, which there's a link here, the Biomart vignette, and the Genomic Features vignette. These are both pretty nice because they go through all of the different things that you can do with each of these packages. So the way this is going to work is we're going to talk about each of these packages, and I'll show you some examples, and then we'll give you a chance to practice. I'll be presenting the first half of this, and then Lori will take over and present the second half. So the packages we're going to use are these 8 or 10 here. So once you load in our studio, once you load the Annotation DBI package, all of these should be automatically loaded. So you shouldn't have to go in and load those by hand. So the goal here is just to be able to show you how you can do some annotation of your data and how the Bioconductor packages interact in such a way that you can sort of get all your data annotated and then put it in a way that you can sort of easily look at the data yourself, or you can present it to, say, your PI and show, here's the gene, here's all the data, and have it be meaningful. So what we'd like to do is we have a couple functions we'll show you how to use. We'll show you how to extract data out of these Txdb and Ncbi packages, which are positional data. And hopefully you'll be able to use Biomart and the Annotation Hub, which, like I said, Lori will cover that stuff. So at a basic level, what do we even mean by annotation? So normally I think of it as, like, for a functional annotation, normally you have some sort of ID. And this can be, like, if you're working with an Affymetrix array, it's just the Affy ID for that probe. And it's usually some sort of cryptic number or numbers and characters that don't really mean anything. It's just something that's unique and identifiable so they can map that to other things and have it be unique. So generally speaking, you've got this ID that doesn't really mean anything, and you might want to link that to a gene ID or the gene name or the gene symbol, or link it to other species, things like that. But you can also think of it as positional annotation, where you've got this ID that can be like an Entree gene ID or an Ensemble ID, and you might want to know something like, well, what's the sequence of that gene? Or how many transcripts does it have? Or what's the sequence for the CDS? Or where are the histone marks? Different things like that. But you can also think of it as, these are two-way arrows. You can think of it as mapping both ways. So you could say things like, well, I've got this Go term, this is gene ontology term of apoptosis. What genes are all considered to be apoptosis genes? So you can map from that Go term through the ID to the gene ID or the gene name. So these are all two-way streets here where you can map from any one of these things to any other thing. So it makes it kind of nice depending on what you've got. So the specific goal that we have is we've got these data, right? And I usually think of it as being this rectangle where the columns are the samples and then the rows are each of the genes. And we've measured that. And so we've got all these measurements. Then we do some statistics on it. So we've compared two groups or whatever. And so we've got a T statistic and a p-value and maybe a fold change. And then we want to be able to say something about what those genes are. And so we want to do some annotation information. And so you can think of this as sort of just being one big matrix of data where you've got maybe the data and then the statistics and the annotation info. And for each row, it's a gene, and the annotation info, you could have like the gene ID and the gene name, and I don't know where it is in the genome, something like that. But remember, so each one of these rows is for each single gene. So that'll come up here in a minute. Bioconductor has got some data containers that make it easy to deal with this sort of data in as much as you can encapsulate all this data in one thing, where you can have the sample data, the annotation data, and the experiment data. So the experiment data would be like your gene measurements and your statistics and things like that. And then you have your annotation data that tells you something about the rows, and your sample data that says something about the columns. So you can have information about the samples, if they're treated, what's the age of the subject, what kind of is it mice or is it humans, whatever. So the sample data thing here tells you about the columns, which are the samples. The annotation data tells you something about the rows, which are the genes, and it's encapsulated in such a way that you can act like it's a data frame. So you can subset it on the columns, so you can subset it on the rows to get different chunks of that without having to worry about, did I pick the right samples? Did I pick the right genes? Because it automatically does that sort of picking for you. We tend to run over time, so I'm not going to go into these anymore. But if you come back, you can look at these examples, and you can open up an expression set and play around with it and look at what's inside of there. So the pros and cons of these. The pros are that you got validity checking. So if you put one of these objects together and you don't do it right, it will tell you that it's not right. It'll give, you'll get an error saying that this isn't a valid object. So that's kind of nice. It gives you the subsetting where you can pretend like it's a data frame and just subset on rows and columns, which is nice. You get function dispatch, so you can do things like there's a show function that tells you sort of this high level what's in it. You can use, if you're going to do comparisons, you can use like the Lemma package. We'll just automatically extract the data that's in that in such a way that in the end, the annotation ends up in your top table. So when you've got output, all of your data that was in there gets sort of lined up nicely in your output. So it's got these nice pros to it. The cons are that, you don't want to create these by hand. It's sort of hard to get the data out by hand, and you can only use it within R. You can't like give somebody an Excel spreadsheet of these things. You have to like extract it. So we have, with our annotation sources, we've got a bunch of different packages that we provide to people. And I've got them listed here in sort of specific to general. So like the Txdb, an example would be a HUGO 20 transcript database. This is specific for a particular Affymetrix array. So it just tells you things about the IDs that are on that array and what genes it measured and things like that. The OrgDB is based on the species level. So this is sort of one level up, where you have like, this is the human organism package. We have ones for mice and rats and lots of different species. The Txdb and the Ensemble Db packages are sort of at the same level as this OrgDB, but they're positional. So it tells you something about where the genes are on the genome, where the promoters are, where the exons are, things like that. The Organism Db is an abstraction of these two, where you can do these sort of cross database joins, which Lori will get into that, but it makes it nice because then you can ask things about positional data and sort of functional data and have it output without having to like sort of link those two things together yourself. The BS Genome packages are sequence packages. So like this BS Genome H. sapiens is the HG19 UCSC version of all of the sequences on all of the chromosomes in the human genome. And so you can use that for extracting out sequence information, like the actual sequences, that is. There's some ancillary ones. This one doesn't exist anymore. I should probably get rid of that. GoDB does all the mappings from gene ontology terms to what those terms are and things like that. And then these last two are online resources that Lori will get into. The Annotation Hub is a way of allowing people who have generated their own versions of these packages to put them on the web somewhere, and then you can just query and get those. And Biomart is a way to query Ensemble resources. So when we're interacting with these Annotation Db packages, the main function is called select. And this is how the function works. So it's select, you have you've got 4 arguments here, the anno package, the keys, the columns, and the key type. So the anno package is the name of the annotation package that you're going to query. So that can be like that org DB or a chip DB, whatever. The keys are the IDs that we know. So if you're working with like a Affymetrix array, these would be sort of the probe IDs from the array. The columns are the values that we want. So we have these IDs that are sort of inscrutable and we don't know what they are. And then the columns are the things that we want to map them to. And then the key type is the type of key used. So we've got the second argument is keys, and key type tells you, well, what kind of keys even are these? So when we're talking about how you can map different ways, you could have a key type of like a gene symbol and map to get the probe IDs if you wanted. So, and if the key type is the central key, it can remain unspecified. So the only arguments that are required are these first 3. So here's a simple example. Let's say we just have some gene ST 2.0 array and we've measured some stuff and we want to get the gene symbols out of it. So if we load the package and we do this for reproducibility, so we always get the same 5 IDs, and then do this line, which will spit out those 5 IDs. And when we look at them, it's just these 5 numbers. And like I was saying before, this is sort of useless information. I mean, 168, 58, 710, that doesn't really mean anything to anybody. So we wanted to say, well, you know, what gene symbol is that? And we want to do it for all 5 of these. So this is how we do it. Select, this is the package name, the IDs are these 5 IDs, and we want the symbol. So we run that function and it spits out this data frame. And we've got for the probe ID column, we've got the numbers, and then we've got the gene symbol, right? So this works pretty nice. I mean, it's exactly what we expect. So questions. First, how do you know what the central keys are? So you don't have to specify them if it is the central key. You don't have to not specify it. You can always say what the key type is. So it's just sort of like if you want to be one of the cool kids and not use this last argument, you can not, right? So if it's the chip DB, the central ID is the manufacturer's probe IDs. So I knew that these IDs are the central ID, so I can just say, you know, I don't have to say that it's the probe ID, right? Sometimes you can look in the name, so org.hs.eg.db. So EG means Entree Gene ID, which is the Ncbi gene ID. So if that's what you have, then you can just use Ncbi IDs and it'll know what you're talking about. You can get examples. You can do this head keys anno package, and that'll tell you sort of what the central keys are. It'll give you some example of them, and then you can infer from that. But you don't have to do that. You can always say, I could have said right here, I could have said probe ID, and it would have done the same exact thing. So the next question is, well, what can I even get out of this thing, right? What can I ask for and what can I get out? So the key types are the things that you can ask for. So if you say the key types for this Hugene transcript cluster DB, these are all of the different things that you can ask for. And there's a page that I forget where it is. There's an actual help page that tells you what all these are, but a lot of them you can infer, like accession numbers, Ncbi accession numbers. Alias is a useful thing because a lot of times the gene symbol that people, maybe you're working with a PI that's old school and he calls it gene, you know, XRF3, and that's changed and it's now called GDF or whatever. So that would be the symbol, the new version of the symbol, and then alias lists all of the things that it's been called in the past. But anyway, so these are all the different things that you can use as inputs, and then the columns are all of the things that you can ask to get out. So like for this one, probe ID obviously is one of the things that you can put as the input because that's the central ID. But you can also ask to get the probe ID out as well. So you could say, well, I've got bracket2, what's the probe ID for that? There is one issue with select, however. So if we do something like this, I just picked these 3 IDs because I know this is going to be a problem. And if we do select with those IDs and ask for the symbol, which is the gene symbol, and map, which is the position, the karyotype position, we ask for 3 things and we end up getting 9 rows. So if you think back what we were talking earlier, where you've got each row is a gene and we want to annotate it, this doesn't work if you end up having 7 rows for the same thing, because you want one-to-one row correspondence. And this got blown out because this probe right here measures a whole family of these DX11 genes that are on lots of different genes, and you don't really know which one it actually measures. It could measure all of them. But this is problematic because we want to have this one-to-one correspondence, and now we've got these extra rows. And if we did this for all of the IDs on this array, we would have a annotation that's got way more rows than the actual data. And then we would have this problem of like, well, what rows do we want to get rid of? How are we going to clean this up so we can put them together the way we want? One way we can do that is use another function called map IDs. And what map IDs does is it gives control of those duplicates. So, like with select here, one thing that we can do is we can ask for multiple things. Well, that was pretty cool. We can ask for multiple things. We can say we've got these 3 keys and we want to get 2 key types out. You could actually have a bunch of those. The problem with that is if there's multiple mappings in here, you can end up with these rows blowing out, which is sort of the problem that we're talking about. Map IDs gives the control of the duplicates, but you could only ask for 1 column at a time. So the arguments for this are the same as select, only now we always have to say what the key type is. So if we do the same thing that we did above, using those same IDs, now we just get 3 things. So, and you'll note that this thing that we got for this 166 ID is the very first one. So this 166 ID, it's DDX11L1. And if you look here, that's the very first thing we got here, DDX11L1. So by default, what this map IDs function will do is it's just going to give you the first one, which you may want, you may not want. That's sort of the naive way to do it. And for a lot of these things, if you've got like 20,000 genes that you're dealing with, it's sort of hard for you as an analyst to say, well, I don't really want this ID to be DDX11L1. I know more about this and I want it to be the third one, right? But in general, like for somebody like me, I don't know that information, so I just take the first one. So there is one last argument here that I've not shown, and it's called multi-vowels. And the other thing about multi-vowels is, so I'm using positional arguments here, right? So I put this one first, and since it's in the very first position, that's the first argument. And then IDs is the second argument, symbols the third argument, probe ID is the fourth argument. There is actually an ellipsis that goes right here, and then after that is the multi-vowels argument. And because of that, we always have to use named arguments for this multi-vowels. So if we were to redo this thing here and we say, well, we want to control what happens with these multiple values, we have to actually say, what the multi-vowels argument is. So if we do this map IDs again, and we say the multi-vowels is a list, we get a list. So now the list IDs are the original probe IDs, and then the list contents are the genes. So you can see like this 166 ID, now we've got all 7 of the symbols in this one list ID. So that's one way we can get things out and then still have one sort of row, if you will, for each gene. Other choices are character list, and this is nice if you're using something like a capital D, capital F data frame, which is something you'll learn about if you use bioconductor stuff more. And it is this thing that looks like this. It's basically a list, but it can be put into these matrix, these sort of rectangular structures. So we say character list, and then we get this thing like this that you can put in a data frame. And then for that, for this particular row of that data frame, you'll just have all 7 of these things there. We can say filter, which is basically saying, look, if there's multiple values for any of these probes, I don't want them because I don't want to deal with it. I don't want to try to have to figure this out. So if we say filter, we only get 2 things back because this DDX 11L1 thing gets filtered out because there's 7 of them. And then the very last one you can do is as NA, and it will do what the annotation packages used to do in the past, which is just to give you an NA. It's not available. And that used to be the default. That's used to be how we did things, and there was a reason for that. And I don't think it's a particularly good reason, so we don't do that anymore. We just give the first one. But there is an argument that you could say, well, I don't want to deal with these duplicates. I don't want to try to figure it out. So I'll just say, yeah, I don't really know what it is. So here's some questions that we can work through. Using either one of these packages, the OrgDB or the transcript package that we've been using, what gene symbol responds to Entrez gene ID 1000? So we've actually got 4 of them here. What's the Ensemble ID for Ppr Gamma? So now we're going to be mapping from gene symbol to Ensemble ID. So this is one of those mappings from the outside of that sort of arrow diagram that I showed, where we're going from gene symbol through the central ID to the ensemble gene ID. And then what's the Uniprot ID for GapDH, which is another one of these. Take one key and map to another key. So go ahead and try that. And I'll give you probably 5 minutes. You can play around, and then we'll show you how you do it if people are sort of stuck. Okay, so let me stop this share. This guy. So the first question is, what gene symbol corresponds to Entrez gene ID 1000? So we've got the select function we can use. So we're going to select using the org.hs.ec.db package. We're going to look for 1000, and note that the keys are always character. So the gene ID looks like a number, but we always have to put it in as a character. If you just put the number 1000 in there, it's not going to work. And we want the symbol. And so it spits out that it's CDH2, right? Pretty easy to do. What's the ensemble ID for Ppr Gamma? So again, we'll use select. Ppr Gamma, and we want the coded. Now in this case, the symbol is not the central ID, so I have to actually tell it what that thing is. So Ppr Gamma is a symbol. So we'll say symbol, and it spits out Ppr Gamma. This is the Ensemble ID. So we can map from ID all the way through the gene ID and then end up getting this Ensemble ID. What's the Uniprot ID for GAPDH? So this is going to be essentially the same thing. This will be Uniprot. What did we say? GAPDH. Right, and then with this one, you see we get the two. There's actually two Uniprot IDs for GAPDH. So if we did something like map IDs, we just get the first one, right? How many of the probe sets from the expression set we loaded map to a single gene? So we loaded, what did we load? Oh, this thing. So if we do this, so this is a way to load things that are installed in the package structure. This is not something that you would in general ever actually really do, but for purposes of doing something like this, where we're explaining, how to do something, having something that's available like this is kind of easy. So then if we say we've got, what's it called? ESET. So we loaded this thing called ESET, and then if you just type the name, it tells you what it is, right? So the question is, how many probe sets from the expression set map to a single gene? So this gets a little bit trickier. So we would want to do something like, so that's the package that we're using, and we want to use the row names for the expression set, and we want to get, we'll use the entree gene ID, and then we're going to use the probe ID to get that, and then so you see what I've done here? So I'm just saying the row names for this expression set are just the probe IDs. And I'm saying I want to get the entree gene ID. I'm passing in the probe ID, and I want to spit this out as a list. So if I've got any things where I'm mapping one probe ID to multiple genes, I'll have a list that's longer than one. So then if we just do supply and get the length, and then do a table on that. What are you doing? So this tells us how many we have and the length. So we've got like 31, almost 32,000 genes that are probe IDs that map to a single gene. And then you can see like we've got 2 genes that map to like 26, 2 probe IDs that map to like 26 genes. So some of these are pretty crazy. They map to like lots of different stuff. So if we go back to workshop, so the Txdb packages are similar to the OrgDB packages, but instead of doing the sort of functional thing where you do the ID to like gene symbol or probe ID, gene symbol or Go terms or things like that that are sort of functional, these map things to positions. And usually the name tells you basically what's in it. So like the Txdb is the Txdb saying it's a Txdb, species, source, build, and table. So like H. sapiens means it's Homo sapiens, Ucsc means it comes from Ucsc, HD19 means it's the GRC H37 build, and known gene means it's the known gene table from Ucsc. And so remember, with the functional annotation, that gets updated on a regular basis. Like so, Ensemble gene IDs change over time, and what symbol for a given gene can change over time. And so the org.db packages are updated every 2 years, and they're based on what was current at that point in time. These positional packages, they instead are based on builds. And so like the build of HD19, this is from like, I don't know, 2003 or something. And so this is more or less a static thing. The functional data actually gets updated, but you know, HD19, they do some little updates, but it's basically sort of a static thing. So if you've got old data that somebody analyzed, you know, back in, I don't know, 2010 or whatever, and you want to recapitulate that, and they did HD19, then you can get all what we thought the positions for all the genes were based on these IDs, on the builds. So the Ensemble Db packages are similar. The difference, the Txdb usually use Ncbi type IDs and positions, and then Ensemble Db packages use Ensemble. These are not particularly interactable because what Ensemble says is a gene can sometimes vary from what Ncbi says is a gene. So if you try to map between these, you can run into problems because they have these disagreements on how many genes, like how many transcripts a given gene has, where they all are, things like that. So these transcript packages, just like the other ones, you can use select and map IDs. So you can say, you know, select this thing, these 2 gene IDs, and I want the transcript name, the chromosome, the start and the end, right? And then I'm telling it that I'm passing in a gene ID. And so it'll be nice, it'll spit it out. It'll say, look, for gene ID 1, there's 2 transcripts. They're on chromosome 19. They start at this position and end at this position. And you can see there's slightly different positions because it's two transcripts. So they, you know, they're like a little bit shifted. And then for gene ID 10, there's just 1 transcript. It's on chromosome 8, and here's where it starts and stops. You can do the same thing with an Ensemble DB. Note here that, like for Ucsc, it says, yeah, for gene ID, there's 2 transcripts. When we do that and ask Ensemble, it says, yeah, no, there's only 1 transcript, and here's where it is. So this is sort of the differences I talk about, where it's hard to do these mappings because Ensemble says, yeah, for entering gene ID, there's only 1 transcript, and Ucsc says, yeah, no, there's 2. And which is right, I don't know. So, but this is not what you would normally do with these things. Instead, what you would normally do is you would use these things called G ranges. And I'm not going to get into this very much at all because we end up going over time when I start talking about this. But basically, you can use these functions where you say, give me all of the data from this package that is of a certain type. So if you say genes, it'll just spit out this G ranges thing that tells you about all the genes. So we've got gene 1, and this is where it is. It's on chromosome 19, starts here and ends there. And it tells you, basically, it's got all of the genes in it. You can split that up. Like if you do transcripts by, you can get, like for 1 in 10 we showed above, that UCSC says there's 2 transcripts for gene ID 1 and there's 1 for gene ID 10. You can see it's just in this list structure. There's other things you can use. You can ask for transcripts and genes and coding sequences and the promoters and the exons. Like I said, I'm not going to get into that too much, but the genomic ranges and the genomic features packages both have vignettes that go into this in depth. So the reason we want these ranges objects is because we can use that to subset things by where they are. So you can say, as just a basic example, let's say you want to know all of the genes that are within chromosome 1 from base number 1 to base number 3 million. So that's something that you can easily do using these sort of information. So, you know, things like what you can do is like this. You can say, if you have a bunch of gene expression changes and maybe you've got differentially methylated CpG islands, you can sort of map those two together because it's a positional sort of information. If you've got a bunch of SNPs and you want to know, well, here's where the SNP ID is, what's the closest genes? I want to know all the genes that are within a megabase of these SNPs, just sort of things like that. And we have another package, another thing called a summarized experiment that's similar to the expression set, where it's the same idea, where we've got this sort of data frame that tells you things about the columns and this annotation data that tells you things about the rows. In this case, it's a G ranges object. And what's nice about that is you can say, well, I just want to subset this and just get the data that's, you know, on chromosome 1 from position 1 to position 3 million. And that's really easy to do. So we have some more exercises. So with the Txdb, question 1, how many transcripts does Ppr Gamma have according to UCSC? And does Ensemble agree? So basically I'm asking, how many transcripts does Ppr Gamma have from Ensemble? And then tell me how many genes are between these two positions on chromosome 2 in the HG19 genome. So basically what I'm asking for is the UCSC, because HG19 is a UCSC sort of a thing.
In the question and the answer, there was one, if it can be used for bacteria.
It depends on the bacterium. There are, I believe, you would know better than me, Lori, there should be some on the Annotation Hub, depending on the bacterium. There is also a function that we're not going to get into called make Txdb from UCSC. So, or make Txdb from GFF. So if it's a species that's on UCSC, you can make one of these things yourself. If you have a GFF, like if you go to Ncbi or Ensemble and you get the GFF, which is just a text file that tells you where all the exons are and things like that, you can use make Txdb from GFF and it will parse that thing and then put it into one of these databases that you can then query. And both of those work pretty well and they're pretty nice. And they're actually pretty fast as well. So let me share this. So we'll go back to RStudio. So question 1, how many transcripts does Ppr Gamma have according to UCSC? So we already looked up here and we know, oh, we don't know. So if we do, I don't know what the idea of Ppr Gamma is. So if we do, okay, so on the symbol Ppr Gamma, the entree gene ID is 5468, right? So we need that information to start with. Now let's do something like, so we want to do so we're going to do transcripts by, and then the stock argument for this is by gene. So we'll just like go ahead and do that. So transcripts by says, give me all the transcripts by something. So you can see here this little pop-up. And so we can get the transcripts by gene, by exon, or by CDS. And so the first here is gene, so we'll use that. So we let that run for a second. So then this gives us it gives us this thing called a G ranges list. And the IDs here, this is the gene ID. So it's gene ID 1, gene ID 10, gene ID 100. So we already figured out that the Ppr Gamma entree gene ID is 5468, right? So if we do, what is it, 5468? So this will spit out the G ranges object for Ppr Gamma, and we can see that there's 10. 10. So there's 10. So according to UCSC, there's 10 transcripts for Ppr Gamma. Now we also did for Ppr Gamma, we got up here the Ensemble ID, which is this, right? So the second question is, does Ensemble agree with that? So we're going to have to do this one. So this Ensemble DB, this is Homo sapiens Ensemble version 79. It's old, but that's just what we'll use. So it's the same exact thing. We get all the transcripts that Ensemble thinks there are, and then we will do this. I think you can copy paste in our studio. You can. So this is what I was talking about before. So UCSC says, yeah, there's like 10 transcripts for Ppr Gamma. Ensemble says, yeah, no, there's 14 of them, right? And these are not all in the same place. So I don't even see much agreement here. They're all on chromosome 3, but you look at the start and the stop, there's not even a lot of agreement on where the start and stop is. So the take home for that basically is, if you start with Ensemble IDs, you should stay with Ensemble IDs all the way through your annotation. If you start with Ncbi gene IDs, stick with Ncbi gene IDs all the way through, because you don't want to get into this issue where you're trying to say, well, what's going on? You know, Ensemble says there's like no transcripts or way more transcripts or whatever. You don't, it's just easier if you just don't do that thing. So then the last question is, how many genes are between 2 positions on chromosome 2 in the HG19 genome? So we want to get the genes. So we just get all the genes for this UCSC thing. So this genes is a G ranges object, and it just says what the start and the stop for that gene is. And really what that is the furthest extent of any transcript for that gene. So the very beginning of the first transcript to the very end of the longest transcript. So now we want to know the genes between these 2 positions. And what we can do is we can make G ranges like this. So it's just G ranges, the chromosome, and then I ranges, which is the start and the stop. And that'll give us this thing. Or we can also do, you can use the Ucsc convention, which is you've probably seen this if you look at the Ucsc browser where they do the chromosome and a colon, and then the start and the stop separated by a dash. So if we do that, it does the same thing. Oh, wait. Oh, sorry. I just have to do it right. Now, so we get the same exact thing, right? And so what we want to do is we want to know how many genes are between these positions on this genes thing. So if we do something like so if we do this subset by overlaps, we're saying subset this genes thing by all of the, by what overlaps this GR2 thing that we specified. And it just spits out that there's just one thing. There's one gene in there, 7260. Okay, does that seem pretty straightforward? So,
There's two things. Can you increase your font a little bit? And then there were a couple questions if you wanted to check out in the chat and the question and answer.
okay. So you want me to increase the font on RStudio, I assume? Let me do that. We're at 16. How about 2? Let me switch back and check. So is that better? Or should I go a little bit bigger? Maybe let's go a little bit bigger. That still fits, right? We'll do that. Okay, and then the other question was, increased it. Where can we find? Oh, so it's the question, can it be used for protein sequence, identifying particular protein responsible for an expression? That's a good question. And the answer is maybe. So that would probably be something you would go to Uniprot to get. And we're not going to talk about that, but there's a package called uniprot.ws that you can use to make queries to the Uniprot database. It's a little bit beyond the scope of what we're doing, but you can use that to get the, I believe you can get the protein sequences using that. So if we go back to.
And then there was a question in the chat too about how to get Uniprot IDs when using the make work package from Ncbi.
See that one?
It's in the question and answers.
Oh, okay. If you make Candida auris, right. So one of the issues with making these annotation packages is when we make the ones that we provide, it's actually a little bit more work than what we can do programmatically. So that make OrgDB from Ncbi, what it does is it goes to Ncbi and gets a bunch of information, downloads that and parses it. And then it also goes and it gets data from 1 or 2 different gene ontology consortiums. but it doesn't do the Uniprot because Uniprot is kind of hard to do programmatically. So to get the Uniprot IDs, you would probably want to use the uniprot.ws package, which you can map between like untrained gene IDs and Uniprot. Yeah, unfortunately, it's hard to do. It's hard to do that with programmatically. Okay, so I'm supposed to be sharing.
Then I guess there's one more in the chat that popped up. Could you provide an example about gene expression changes in your differentially methylated CPGs?
Right. So if you wanted to do something like that. So am I sharing my screen? What's going on here? Lori, am I sharing my screen?
I don't see it yet, no.
What the heck? Oh, here we go. I minimized that. Okay, so doing something like that would be, so if you had differentially methylated regions, what you would have is you would have a G ranges, right? So you go through, use like a methylation package, and it'll spit out basically a G ranges. So you'll have something that'll be sort of similar to like this G ranges list right here, where you've got for each differentially methylated region, it'll say like it's on chromosome 2 from, 10,000 to 10,050, whatever, right? And then you also would have the genes. So you've got these genes where you know where the genes are. So then you can do this overlap where you can say, you can subset the genes by the overlaps, or you can subset the genes by an extended overlap. So there's ways that you can take, like if you've got the G ranges from the methylation regions and you want to say, well, I want to know all of the genes that are within 1 megabase, there's ways you can take and say, take those G ranges for the methylation and extend them 1 megabase, and then give me all the overlapping genes that overlap the extended ranges for each of those methylation peaks. Or you can say, well, I only think it works downstream, so you can take those methylation ranges and extend them one direction or the other. That's a little bit beyond the scope of what we're doing here, but that would be something you can find in the genomic features package. So let me share. So a new package that we're producing now is an Orthology package. So the nice thing about this is if you need to do some orthology where you've got like, if you've got one species and you need to map it to the other. So like this example here is we're going to select from the Orthology DB. We're going to use some keys. These are human gene IDs, and we want to get zebrafish results out. So if we do that, we end up getting 6253 mappings. If we get rid of the NA mappings, we have about 10,000 mappings. And so what we end up with is this data frame where we've got the Homo sapiens gene ID and the zebrafish ID. So this just uses Ncbi data to do these mappings between the two species. So, I mean, that's kind of nice, right? There's 368 species in that package. There's like a lot. I mean, you could do like rhinoceros to human, whatever you want to do. So Organism DB, this is where Lori takes over.
Although there were a couple questions if you wanted to look at them. I mean, I think we still have a full hour slotted. So if you wanted to look at those.
How do I align Ensemble gene IDs to the how to align Ensemble gene IDs to org? Okay, so this is something that I actually argued against doing. So, okay, so the question is, can I go over how to align Ensemble gene IDs to org.rn.et.db genes? So let me stop that share and we'll start this other one. So the question is, how do you map Ensemble gene IDs? So you can do this. And so like if we do, we're going to get a bunch of so let's see, do we have that? Can we install on this thing? So I'm just going to install the rat org.db package, although I probably could just use whatever. So we load this rat package. And so normally we would get these IDs elsewhere, like, right? So you've got a bunch of Ensemble IDs and you want to map those to, what are we mapping them to? Okay. So let's say we've got a bunch of Ensemble IDs and we want to get the matching Entree gene IDs, right? So we'll have so we're just going to get all of the Ensemble keys out of this thing, right? So then, so right, so these are all Ensemble rat IDs. And the question is, you know, how would we map those to like Ncbi gene IDs? So one way we could do this, so we've got 21,374 of those, right? So if we do so we're going to select from that rat package these IDs I called Ensemble DB, which whatever. We want the Entree gene IDs, and we're going to use Ensemble IDs as the input. So you get this message saying select return to one-to-many mapping between keys and columns, right? So we know we have 21,374. So if we do rows of the Z object, so we have some duplicates here, right? Because there are some genes where Ensemble says, yeah, I just have one ID for that gene, and Ncbi says, no, we've got two for it, right? So we could either accept that, right, where we do so if we do like so to get those out. So we've got this. This is our thing, right? So we can see right here. Here's one of the deals. So Ensemble Entree Gene ID. There's two here that point to. I mean, this is the same thing. 24,153, and it points to two different Ensemble Gene IDs, right? And so there's probably going to be issues going both directions where we've got single entree gene IDs saying that it points to two different things and vice versa. So Assad, you raised your hand. Can you just type in the Q&A?
Okay, I think I was given the privilege to speak.
Okay.
So I wanted to ask, instead of having entree IDs, which gives you like numbers here, what if we want the actual gene names? What do we do differently?
Right. So you want to get the gene name for Ensemble IDs? Is that what you're asking?
Yes.
Okay. Right. So in that case, we would do essentially the same thing, except for instead of asking for the entree gene ID, we can ask for the symbol. Right?
Oh, I see.
Right.
And
there's also that. So you can either get the symbol or the gene name, or you can actually get both. You could say, give me the symbol and the gene name. Does that make sense? Cool. But yeah, you can see. So what ends up happening, it's like there's these two, right? So there's two gene symbols for this. There's one gene symbol for these two genes. And the way all these annotation services work is people provide data to the annotation service, and then they end up over time, probably what's going to end up happening is these will get combined, right? So Ensemble will end up saying, oh, look, this is really the same thing. So we're just going to combine those two things. Or, maybe they won't. I don't know. So if we do like, we don't have it. I'm not going to install it. So we could look and figure out why that is, but cannot find. So you're using a Docker or you're using RStudio? So you shouldn't need to pull stuff from Docker for this. You should just be able to do, if you do, So if you do this, library Bioc 2021 Anno, you don't need the Docker. You just do, if you just library Bioc 2021 Anno, that will load like all of these packages. It should.
Or if you're not using the Orchestra app, then you would need to do install of them first. But
so you could, if you're not using the Orchestra app, if you're just using your own, what you could do is you can do that. So as a side note, anytime you're installing anything on R these days, you should just use Bioc Manager, because it's like it'll use anything that it needs to in order to get the installation. So like right here, I'm saying install, and basically what I'm saying is go to my GitHub and get this thing. And so what it'll do is it'll just like automatically know, oh, you just want to go to J MacDon's GitHub and get this thing that he's got there. So, it's kind of nice. I mean, it's using a different package under the hood to do that, but you don't have to know, oh, I'm using this package or that package, whatever. If you just use Bioc Manager and install, you can just install anything you ever want. You don't have to worry about things. So yeah, so one of the questions is, what do you do when you've got duplicates like this? And that's a good question, and I don't have a ready answer. So basically, the reason they're duplicates is because at this point in time, or at that point in time, Ensemble, so this, you have to understand too, this we're using an old version of Ensemble, or no, never mind, this is new. So for whatever reason, Ensemble right now has a mismatch with HGNC. So these symbols come from the HGNC. And so Ensemble says, look, there's these two things that maybe they're on a different place, different position. So if we do, so we can take and like get the position. So we'll get the position information for these two things. So basically what I'm saying is, I want these two things, and I want the map, which is the map is sort of the karyotype position, the P and Q business, and I'm going to use Ensemble to map it. So it doesn't have any position information in it. So here I'm saying, just give me all the Ensemble IDs from the human one, and we're going to get the symbols, right? So it's basically the same thing. So if we do, no, it's not install packages. you load. So somebody asked a question, they said, this isn't going to work. Install.packages is a completely different thing. You want to load the Bioc Manager package and use you can do. So this is the fully qualified function name. So this will make sure that you get the exact one that you're looking for. So Bioc Manager :install will automatically do this. If you try to use the base function called install.packages, it's not going to work.
And the kicker is making sure that you have Bioc Manager installed, and that you do through install.packages Bioc Manager if you don't have it downloaded yet. Right. So the Bioc Manager package is actually on CRAN. And then when you load the Bioc Manager, the install function in Bioc Manager will install anything from Bioconductor or CRAN, or if you get a GitHub link like James just did.
Yeah, so like I was saying, in general, if you're going to install a package, you should just use Bioc Manager. So, So as an example, we've got, here's the deal where we've got like at least, what's that, 6 Ensemble IDs that all point to the same gene symbol. So one thing we can do is so we have the transcripts by for the ensemble, right? And then so we will do oops. So now this, I'm just getting these ensemble IDs. And so if we look at this, so we've got 6 because there's 6 duplicates here, right? And the reason we've got these duplicates is, you can see here, these are out on these different haplotypes. I can't tell, right? So the seq names here tell you what the chromosome is with all the genomes now. Pretty much they have these haplotypes where they're not able to position them quite accurately. So this is one of the chromosome 6 haplotypes. So this gene happens to be on chromosome 6. The problem is this is one haplotype. This is a different haplotype. Right, so Ensemble is saying, if you've got this thing that's on this haplotype in these positions, it's this particular ensemble gene. If it's on a different haplotype, it's a different gene. I'm betting that if you looked at Ncbi, it would be like, no, those are just haplotypes. It's all the same thing. It's just unplaced, and it's probably these positions probably aren't even that different. But anyway, so the point here is that how many genes you have and where they are and what they're called is a very technical thing to do. And you've got two groups. You've got Ncbi in the US and you've got Ensemble in Europe. And they're trying to do the same thing, but they're not doing it the exact same way. And since they're not doing it the exact same way, you can get disagreements. And those disagreements usually end up being something really technical and different for every single gene. And so it's very hard to try to figure out, oh, why does Ensemble say that this is 3 genes and Ontology and Ncbi says it's only 2? Mostly people that are sort of analyzing data don't care about those things. They just want to know, look, where's Ppr Gamma? I don't care about your argument about how many different ones there are and where they are. I just want to know, where is it, right? And in that case, it's easier just to stick with either using Ensemble or using Ncbi and never try to go between them. Never try to convert an Ensemble ID to an Ncbi ID or back and forth or anything like that if you can get away with it. Assad, you've got your hand raised. Oh, are you asking about, can you map the gene locations on a rat genome? Is that the thing? The answer to that is yes. There is a package called txdb.r.norvegicus.ucsc.rn6 or whatever. Oh, it's a different question. You want to type it or you want to, I don't know how to make you where you can speak. I can invite. Okay, you can talk now.
Thanks so much. So I had a question. It's I think it's better if I speak. So I did a RNA-seq differential expression analysis.
Yep.
In a sample where there were 5 sham and 5 traumatic brain injury samples.
Yep.
And at the end of the day, we found some genes to be, I mean, their p-value, I used Txdb package, so their p-value was significant. Now, I just wanted to ask how can, so I have the Ensemble IDs and I tried to convert them and get the exact name. I just wanted to ask how can such data be, I mean, translated into a paper? Like, should I find all these genes and like speak on them in addition to making the plots? That's one question. The other question was—
Oh, wait, can you back up and restate that question?
So the first question is, so to be able to write a meaningful paper,
right?
Do you think it's a good idea to find these genes that are where the p-value after doing differential expression analysis was less than 0.05 and like talk about them, like maybe list them and say these were the genes that were significantly different?
Well, I mean, right. So that's a little bit off topic, but just briefly, that's generally what people do, right? So you end up having the situation where you said, okay, I had 20 different genes or 50 genes or whatever that were differentially expressed. And maybe you'll have a table and you'll say, here they all are. And then maybe you'll then say, and here's some information about these 4 that I want to highlight because I think that they're more interesting in this situation than these other ones or whatever.
I see.
Does that make sense?
Yeah, it makes sense. Okay, I'll look that up. Secondly, there were some genes where when I matched them with Ensemble using the org package, they showed up as NA, but they were significant. I mean, in terms of p-value, what to do in such a
so that's the thing that I'm talking about. So the org packages are all based on Ncbi IDs, and you've got Ensemble IDs. So you don't even want to use an OrgDB package in that context. You want to use, in that case, probably Biomart. And that's something that Lori is going to talk about and how you would use that. So basically the short story is the OrgDBs are where we've taken and mapped all of the Ncbi IDs to all these different things, right? And so if you've got Ncbi IDs, that's the thing that you want to use. But if you've got Ensemble IDs, you don't want to use that, because then you end up with this problem where you're trying to, where you get things like what you're talking about, where it says NA. Well, that's because for whatever reason, NCBI says that's not a gene, and Ensemble says, yeah, for sure it is a gene, right? And so you don't want to get into that argument. You don't care about that. Who thinks it's a gene, who doesn't think it's a gene? You've got a gene that looks like it's differentially expressed, and you've got an ID for it, and you want to say, well, what is it? And in that case, you would use Biomart, which is a way to query the Ensemble database and do that. So you'd use Biomart to say, here's all my IDs, and I want all the gene symbols, right? And so Lori's going to go over that. And
got it. Thanks so much. It's all clear. Thanks.
I don't remember the name of the package too, but there's some in an Annotation Hub that Johannes Rayner generates for the Annotation Hub per Ensemble release 2 that has a lot of Ensemble information in the hubs.
Right.
Yeah, and
so
I say it's like HENSDB or something like that.
Oh, really? Is that a? Because I know he was doing all the Ensemble DBs, but he's got one that's just basically a functional one.
I'm not sure. I'm not sure what's included in there or not, but I know that it's based on the Ensemble data, so it might have more information there.
Right. Okay. So you want to take over?
So can you see the script and our studio session here? Is my screen sharing?
Yep, we can see it.
Perfect. So as you can imagine, that sometimes you want both functional and positional annotations, or you want to determine like one based on the other, and you don't want to have to use the two separate packages and map between the two. So is there a place where all of this information is in one place? And the answer is yes, but for some species and in some cases, but not necessarily all of them. So there's a couple different options for this. The first one are these Organism Db packages that contain all the information in an OrgDB, a Txdb, and GoDB package all in one spot. And it's nice because while it provides a default loaded object here, that's based on particular information. So once we look at this, you can see what information was provided. So this was made with the HG19 known genes Txdb with the default OrgDB, HUGO DB, and the GoDB ontology. But if you wanted to change this to a different Txdb, like the HG38 version, you can swap it out and easily update the object to use a different Txdb object. But all of those same functions that James went over, that you can use with the OrgDB and the Txdb are available here. So you can use the columns and the key types, your selects, all of those same functionality are available here, but all the information is in one spot. And so you could see what information is available by, again, doing what you can query by doing the key types on the object, or key types, or what information you can give back. You can look at the columns and use those in your select calls and mapping calls. again, here's an example with the Txdbs. There's the genes and the promoters and exon functions where they have automatically pulling out those mapping locations to known genes or promoter regions. And those are all available on these Organism Db packages too. So just as a proof of principle here, if we grabbed out and wanted the columns for the Ontology ID, all of the gene aliases and the Uniprot ID, and we wanted it across the genes table, we could look at that, get a warning message, and then grab our G ranges objects with the genes that were overlapping in that region and give the information for the genes available, the Uniprot IDs available, and the Ontology ID. So just as a proof of principle and to reinforce what we were doing with the OrgDBs and TxDBs, there's just a few little Organism DB exercises here. So maybe I'll just give you guys a minute to think about these 4 questions here for get all the Go terms for the BRCA1, and what genes does the Ucsc transcript ID map to, this particular ID? How many other transcripts does that gene have? And then get all the transcripts from the HG19 genome build along with their Ensemble gene ID and UCS transcript ID and gene symbol. So I'll give you guys just a brief minute here to think about those and start thinking about how you would do them, and then we'll go over them. And they kind of progress in difficulty too. So the first one, for get all the Go terms for BRCA1, it's as simple as the select call that we were doing with the OrgDBs. And our term of interest is BRCA1. And then we want back Go term and symbol. So we're giving it, again, to go over, so we want back the Go terms, and we're giving it the gene symbol BRCA1. And if we do that, we'll get the results for all of the Go terms for those samples. For the second question here, what gene does Ucsc transcript UC002FAI3 map to? Again, using our select function, we can do a select on Homo sapiens, and we're going to give it that transcript, or the transcript ID. And this time we want back the symbol. And we were giving the function a tx name. And so this transcript actually maps to ZNF23. How many other transcripts does that gene have? There's a couple different ways that you can do this. There's a transcript by function. And we can use that on our Homo sapiens and then subset it. So the transcript by is going to give all of the transcripts, all of the transcripts for gene, but we want to limit it to that, the transcript that we had in question. So then we can subset it, and we can do a map IDs on the Homo sapien and give it that transcript that we had. And the transcript by is by Ontology ID. So we're going to need the Ontology ID given back. Did I spell that right? Hope I spelled that right. And we were giving it the TX name. So we're getting the mappings so that we can get the Ontology ID in question. And we're getting those results back. And then we're going to subset this transcript by to know how many transcripts were for that given transcript. So there were 6 different ranges. And then the last, was that the last question? Oh, I guess I skipped over. Sorry, I guess I did them in the wrong order. So this was for getting all of the transcripts from the HG19 build along with our Ontology ID and symbol. So how many other transcripts does that gene have? I guess was the mapping of the symbol. So sorry about that. So it's kind of useful to have all of the information in one location, but the only limitation of these Organism Db packages is that Bioconductor currently only provides 3 different Organism Db packages. So just if you guys aren't all aware or have been to the Bioconductor homepage, in the Bioconductor homepage, if we click on the software, it gives an overview of all of the packages that are available in Bioconductor. And there's a list of software packages, but we're dealing with the annotation. And there's this package type. And if we look for Organism DBs, so unless you're using human, rat, or mouse, you can't use an Organism DB that's already generated. So there's another option, which is using an Organism dplyr package. So the Organism dplyr packages extends it out to a variety of different species. And again, it's combining the Txdb and the OrgDB and the GoDB information in one type, just like the Organism DB packages. You can use the key types, the selects, the exons, the promoters, all those familiar functions that we've been going over. And it's all in one place. And then if you're familiar with the crayon and the tidyverse, it has nice dplyr-like output and function. So the main function of Organism dplyr is actually this src organism function. And just briefly checking out the help page for this. So there's an src UCSC for ones that are specifically UCSC, but most people like using the src organism. And the input is a Txdb, and you can get all of the organisms that are supported using the support organism function. So normally you would give it a Txdb, whichever Txdb that you wanted to work with. There's a couple helper functions in here that give a pared down version just for size and speed of loading and building database. And if we look at this src object, we can see where it's generated and then what sort of tables are available, like ID of session number, ID go all. So these are all different tables that have various information, range CDs, range exons, range genes, all of the information acquired from the OrgDB and Txdb of interest. And again, as proof of principle, you can use your promoter function on the object and get promoter regions, exons, genes that were in the OrgDB. If we wanted to see what information is in one of these tables to see what other information is available, we can use table function on the src object and give it the name of the table. So if we were just looking at ID, we can see that it has an Ontology ID, a mapping, an Ensemble ID, your symbol, your gene name, and any aliases. So you can start thinking of making complex queries with just using that one object. So thinking about this query that we have here, and this inner join. So basically what I was looking for here is I wanted information on the gene symbols for ADA and NAT2, and I wanted their position information along with their alias and map symbol. So basically what this inner join is doing is saying, I want to join the tables for the IDs and the ranges for the genes, which is where the information that I need to do the filtering is. I'm going to filter it down for the 2 genes in question for ADA and NAT2. And then I'm going to select the columns of information that I actually want to display. And that's what this table here is depicting here. So you have your chromosome, your start, your stop, your strand information, and then your symbol, your alias, and your map information. So I have some Organism dplayer exercises here. So again, maybe just give you a minute or 2 to think about these, and then we'll go over the answers. So how many supported organisms are implemented in the Organism dplayer package? So for that one, I mentioned that there's this supported organisms function. And so that will give you all of the different organisms that are available in Organism dplayer for use. And how many are supported? We can do, well, we can look by the table there, but if we just get the number of rows, so there's 21 supported organisms in Organism dplayer currently. So using Organism dplayer, display the Ensemble ID and gene name description for the symbol NAT2. So again, basically can be accomplished with a simple select. So if we do a select on our Organism dplayer object and we give it NAT2, and we said we wanted the Ensemble ID and the gene name. So there's 2 different columns there. So we want the ensemble and the gene name. And we are giving it a symbol. And so we can get the ensemble and the gene name. Show all the aliases for NAT2 in the database. Same kind of concept. We already have this, so it would just be changing it to alias actually. You can get all the different aliases for NAT2. So the bigger one, so display the gene ontology information for the gene symbols in NAT2. So again, if we look at this SRC, we can see that we have a couple different options here for Go information. We have IDGo and IDGo all. We'll just concentrate on the IDGo table so we can see what information is available in that IDGo table. So we have evidence and ontology in the Go term mapped with the entourage ID. So we can make one of those inner joins. So if we do an inner join on the SRC ID table and the SRC IDGo table, and then using our dplayer methods, we can send that as a pipe into a filter, and we can filter for the symbol that we want. So in this case, we said that we were going to look for NAT2, or that we wanted information on NAT2, and then pipe that along into select. Because there's a couple different selects going on and we have the dplayer, we'll probably want to specify to use the dplayer select method so it doesn't get confused with the select method for the org DBs. And we're going to select entourage, ensemble, symbol, go, gravity. the evidence, and the ontology. I think I had to put all of those in quotes, didn't I? And this will give back a table with the IDs and all of the Go information that we wanted to include using just the one table. So it's kind of convenient to have all of the information in one place on one object. So another type of annotation resource to consider is the BS genome information that provides sequence information. So the main function in, well, I guess first, so there's all of the available genomes. So there are a lot of genomes available, and I encourage you to search this. So let's see, available genomes. Let me get the dimension of it. No, because it's a list. So there's 106 available BS genome objects by default. We'll look at the BS genome for humans using the Ucsc information and HG19. So we can load that in. The main function of a BS genome object is a get seek function. And you can search a BS genome object either by full chromosome, or you can give it a G-range object or a G-range list object. So you can search for particular regions of interest. So in this case, we searched over the entire chromosome 1 information. In this one, this GNS was taken by using the genes on a Txdb object. We had run that earlier. So if we look at GNS, these are the known genes from the Txdb object. So we're taking a particular gene region and getting the sequence for it by using that filter. So just as a little back and forth and using all of the concepts, can we get the sequences for all the transcripts of the TP53 gene? And how would we accomplish that? Well, thinking about it a little bit. So we need to get our transcripts. So we need a transcripts by our Homo sapiens object. Because we need to get the transcript information for this TP53. So let's save that into an object because it'll be a G-range list. So then we're going to map the IDs of our Homo sapiens object, and we want the TP53 gene. And we're going to want entourage, if I can spell it right. I always forget the T. And we're giving it the symbol. So this will give us the transcripts for the T53 and the G-range information. So then using that get seek, we can use our BS genome object and put that TP53 in and get all of the sequences for all of the transcripts for that gene of interest. So the other really good resource that Bioconductor has is we're getting into some like online and interactive resources now. So the first is Annotation Hub. So Annotation Hub is nice because it's user contributed. So the Bioconductor core will provide some resources in Annotation Hub by default every release. And then we really rely on the community to add annotations into the Annotation Hub if they think it will be useful for distribution to the community at large. But to use Annotation Hub, we first call the constructor of Annotation Hub. And it's going to download this database of meta information for all of the available resources that you'd be able to download. And if we look at it, so currently there are 60,134 available resources in Annotation Hub. And the thing about Annotation Hub is it's really about the queries to find the right information and what information is available. So there's this idea of metadata columns that when someone submits data to the Annotation Hub that we require certain information about it, and then those M columns become queryable through the database. So what kind of metadata do we have? We have things like the title of the resources, a genome, data provider, description, species, the R data class. So thinking about queries and what's in there, like we could look at what data providers are available. So if you want Ensemble information or UCSC information, stuff from the Broad. So you can query based on data provider. You could query based on the R data class. So maybe you like working with the G ranges objects or 2-bit files or these Txdb objects and what are already created in there. Or of course you could search by species. So those are always an option. And there's this query function on the hub that then you can paste together different query options. So again, maybe we liked G ranges, and we want human, and we want ensemble information, and we can query to see what resources match those query terms in the hub. And there are 100 records that match those search queries currently. And then if we wanted to look at one particular resource in particular, we can use this AHID. And if we put the AHID in with the single bracket, it will give us all of the metadata for that record. So we can really query and look at resources before downloading them to see if they're the resource that we want. So in this case, when we find a resource that we want, we can download it with the double bracket method. So in this case, we have what I want, and we query it, and we want to download it. So we use that AHID with the double bracket, and now it's downloading it to our local system, or in this case, our studio session, and importing it in R. And because we chose that G ranges object, we expect to see a G range object back. Eventually, hopefully it imports. Not exactly sure why it's taking so long, but ideally we get this G ranges object back, and then looking through here too, we can see that then we could convert that into our well-known object that we've been working with the entire time, the Txdb, by using this make Txdb from G ranges function. Great. So if we look at what I want, it's the information from Ensemble for a G range object for Homo sapiens. And then again, if we wanted to, we could convert that into a Txdb object by using the make Txdb function. And then we would get this Txdb object that we could work with the same way that we've been working with. So some exercises with the Annotation Hub. So how many resources are there for, I just picked a species, so salmon, Atlantic salmon. Probably should not have run that. Whenever this gets done. So basically to look up the species for Atlantic salmon, we would just do a query on the hub for that species of interest. And it would give us how many resources are available for. There we go. So there's 47 records that are related to this species. And get the most recent ensemble build for the domesticated dog and make it into a Txdb. So again, we can query the hub for our species and some other search terms. So we wanted dog, so Canis familiaris, I think I spelled that right. And maybe ensemble, and we'll do a GTF. Oh. I wanted to assign it. So this will give us back our query information, and we can see that this age 79003 is the most recent. So we could download that and then turn it into a Txdb. So to download, we would do dog with the age 73, and then put that into a make Txdb from G ranges to get our Txdb. Since it's taking a little while, I won't run that, so we can briefly talk about Biomart. So the Biomart package can be used to connect to various biomarts. We're mostly going to focus on the most popular case, which is connecting to the Ensemble database right now. So if we load Biomart, list biomarts, we'll list all of the available marts available. And if you give it a given URL, so like if you know a given URL, I think there's like a worm-based one and a proteomics one, you could give it the host URL and it will list the available marts. Those are designed to be used with a use mart function. But again, since we're going to focus on Ensemble because that's the one that's most popularly used, there's a use ensemble function. So we can kind of skip over that and go into the use ensemble, and it will give you mart function. Once you have the mart selected, you need to select a data set that's available. So we can use a list data set function to see all of the available data sets for a given mart. Here I just printed out the first 6, but there are many, and sometimes that can be a little bit unwieldy. So if we had an idea of what we wanted to search for, like we knew we were interested in Homo sapiens, we could use a search database function and give it the mart that we are interested in using and a pattern. So maybe we're interested in H. sapiens, and it will give you the data sets relating to that search term. Once we decide the database that we have selected and the data set that we want to use, we can reinitialize the mart object with both the mart and the data set to give us an object, a mart object that then we could query and utilize. And the one thing about Biomart that we've realized over the years is that unfortunately we can't control the connectivity to Biomart. And it seems like right now it doesn't want to connect. What we mostly say in this case is that it's kind of beyond our control and we have to wait for it to connect. So now we have our MART object, and it will try to circle through a couple different nears to see if one is down. It will try a different near because there might be temporary internet connectivity issues, but hopefully one of the nears will work. So in this case, it looks like, I'm not sure what one was used after this, probably US West, but the main function for doing queries on MART objects are this get BM function and you give it attributes, which is what we want, a filter for what we have, the values of the IDs that we have, and then the MART object to use for querying. And there's some helpful functions in Biomart like list attributes and list filters that give you the possible options for kind of like similar to the key types and columns that we were using before for our selects of what would be available to put into your query. So in here we have like a sample query of maybe we had the AFI IDs and we just chose 3 at random. And we could use, say what we wanted was the symbol and we want it mapped to those query IDs. So we want both of those back. We have the AFI IDs, we give it the AFI IDs and we give it the MART, and then we can give our query for the symbols matching to those IDs. So I think we're at time, it looks like a little over, sorry for going a little over, but we have time. I suppose James and I could take any additional questions.
There are some questions in the Q&A as well as the chat. So Hedia Tahani has 2 questions, I think, saying how to convert from masculus to Homo sapiens using Biomart. And the second one is, can the packages be updated using databases REST APIs?
So we do plan to update the packages to use more of the REST APIs. It's kind of a work in progress from the core, especially the core related ones. We're really trying to update many of the packages to using more of the REST APIs to make them more functional, more stable. So it is a work in progress. Converting mouse to Homo sapiens. I'm not exactly sure. I have to admit, I know Biomart, but I'm not an expert in it. But if you want to pop me your email address, I can certainly, and if anyone else is interested in that, we can ping the Biomart maintainer on how to use that, because he would be much more skilled at giving you an eloquent answer.
Plus, there's an example in the Biomart vignette. That's a pretty common use case.
Perfect, thank you. As far as packages optimized for high memory computing environments, HPC clusters, and cloud infrastructure, I think, I mean, as far as optimized for them, I mean, most of them are downloading and querying sources, so I don't see why they wouldn't be. And I know there's been large steps in the cloud front and computing environment with packages like the Anvil package and Bioconductor Parallel to take more advantage and use of clusters in high computing environments. Blast NR package. I'm not sure if there's a Blast NR package, honestly. If there is, it might be by, yeah, if there is, it would probably be in CRAN, but it wouldn't be on Bioconductor or Bioconductor Core 1. Our main ones are this BS Genome. Biomart vignette is broken. Well, I can find a link to vignette that works if you give me 1 second, or hopefully a vignette that works. So for Biomart vignette,
It's not broken for me. Yeah, they're both there.
But regardless, there's the Bioconductor package release page. And if you go down into the documentation section, it will have HTML code as well as all of your code truncated out into an R script for easy use on the package landing page for Biomart. And I just checked, and those both should be active and live and generated. So.
All right, are there any additional questions? I see somebody just posted increasing timeout or downloading by hand.
James, I don't know if you have a good one for this. How can I differentiate the different types of IDs? Available examples are the popular symbol, Ensemble ID, or Ncbi ID.
Differentiate. Well, so the main difference between all of those is where they come from, right? So the symbols come from, well, they come from a couple places. So the symbols originally came from people just naming their genes. Like, so back in the day when they first started, back in the day when people were sort of identifying genes, they would give them a name or a symbol, right? And these days there's a consortium called the HGNC that's sort of in charge of making the gene symbols as consistent as possible. And they plus minus do a pretty good job with that, but it's, none of these are perfect. Like there's duplicates in Ensemble, there's duplicates in Ncbi IDs as well. But so the symbols are useful for biologists because usually they don't know the Ensemble gene ID and they don't know the Ncbi ID, but they do know like BRCA1 or GAPDH or things like that. Those are, for a biologist, those are usually the things you want to use because they're sort of informative. The Ensemble ID and the Ncbi ID, those are really only intended to be identifiable. So the idea is that a given Ensemble ID or a given Ncbi ID is unique, and it also points to like the correct thing. So usually you don't use those for anything other than like identifiability. So you don't have mismatches kind of a thing. So there was, can the packages be updated using database REST APIs? So the vast majority of the packages that we're talking about right now are like these, like the OrgDB and the Txdb and things like that. Those are produced for every release. So twice a year, these things get produced. And they are not meant to be updated really there's two trade-offs. One, like I was talking about, the functional annotations, those change every week. Like the gene IDs get updated on Ncbi every week. They like parse them and like get rid of some or whatever. So there's this idea that those sort of annotations are moving rapidly through time. They're changing over time. And by having every 6 months having these releases, that gives you sort of more updated information, but consistent for those 6 months, right? So it's kind of hard to end up annotating your data if you used what they had on November 16th, and then 3 months later you're updating and maybe things changed, and then it's sort of hard because you have this sort of moving target. So by having the releases, the idea is that the data are updated as much as possible while still remaining consistent for some periods, if that makes sense. But that said, they aren't updated using REST APIs. They can be updated by doing, like, you can use make OrgDB from Ncbi. I wouldn't actually recommend doing that because it downloads a lot of data and then it parses it using R, which it's pretty slow. I mean, it takes probably 3 or 4 hours to build one. So if you can get the org package from Annotation DBI, from the Annotation Hub, or if you can get it from the Bioconductor website, I would in general recommend doing that. Any other questions?
There's another question in the chat about asking about the header for the section we are at now.
I don't know.
As far as the workshop, and I think we're at the end of it. So.
Yeah, we're at the end of it.
And then I think this will be our final question.
From Kingsley.
Yeah, the genome variations.
So in general, so there's competing interests with the two different genome builds. A lot of historical data, I mean, a lot of data is still based on GRCH 37, which is HG19. And so some people tend to default to using HG19 just because, like, all of the ENCODE data, a lot of the data you can download from online, all the HapMap data, you know, a lot of it is all based on HG19. So if you're working with data, particularly genomic variation data that's based on HG19, you can always lift it over, but that's So you can lift it over, or you can just stay with HG19. All things equal, I think that using GRCH38 is the smarter play, just because it's a more recent, and they're actually still, well, actually they're not even working on that anymore. So it's static as well, but it's a more recent static version of the genome. So I tend to default towards GRCH38 just because it is newer. There's actually an even newer genome that is being built where they're using just one individual, and they're trying to do a better job with a lot of these haplotypes that are misplaced. There you go, CHM13. So, I mean, I don't know how much data is available for that one yet, but yeah, there's always this tension between what data you have that's existing and what data you can get. So, oh, it doesn't have the Y chromosome. Yeah. Oh, probably because that's a woman, right? So so it's the CHM13. They did super deep sequencing on one person, because we've got this issue with all of these genome builds where you have all these haplotypes. I mean, if you look at GRH38, there's, I don't know, probably 300 or 400 haplotypes that are just, they're not placed yet. And so one of the common things that people do is there's a function in Bioconductor called keep standard chromosomes. So if you've got, like if you get all the genes, if you do like transcripts by or genes or exons by whatever from a Txdb object, and it's the HD38, you'll have a bunch of data that are on these unplaced scaffolds. And one of the common things that people do is use a function like keep standard chromosomes that basically throws out all of those unplaced haplotypes because they don't want to deal with them. So the idea with CHM13 is to help get those things placed to where you don't end up throwing away a bunch of your data just because you don't want to deal with it. Okay.
Right.
Oh, go ahead.
Now, I was just going to say, I think this concludes our session today, unless you have from any other comment, James or Laurie.
No, just feel free to reach out with any follow-up questions to emails and our Slack, or you find us. We like answering questions.
Yep. Or on the support site. The nice thing about the support site is it's sort of a historical document to a certain extent where people can go and search for already answered questions.
Great, thanks. Thank you so much, James and Laurie, for leading the session today. I can see it was very informative, and I also want to thank the attendees for their attention today. And I also want to remind them about, for those who registered for the session tomorrow, that session is called the Introduction to Matrix Factorization and PCAs. And if you registered for that session, you would have received a link to join the Zoom webinar. And the session will start at 2 p.m. CAT. All right.
Great. Thank you so much.
Bye, everyone. Thank you.
Thank you. Good evening or good morning, depending where you are.